Our next speaker, who I think dovetails perfectly with Christine's talk, is Professor James White. And James will be talking to us about the science behind the relationship between plants and microbes. And James is Professor of Plant Pathology at Rutgers University, New Jersey, and he specializes in symbiosis research, particularly looking at endophytic microbes. These are bacteria or fungi that live inside the plant tissues in a symbiotic or mutually beneficial relationship. James received his PhD in mycology, which is the study of fungi, from the University of Texas and is prolific in his field, having authored more than 180 articles, as well as being author and editor of reference books on the biology and taxonomy of fungal endophytes. And in his lab, the ultimate goal, he says, is to develop new and effective ways to protect plants from biotic and abiotic stresses and enhance the nutritional status of crop plants through construction of functional plant microbiomes with defensive and nutritional properties. And it's their belief that plant microbiome technologies may one day reduce or replace chemical inputs currently and problematically, as Christine explained so elegantly, being used to grow crops and to protect crops at the moment. So delighted to introduce you, James.
Thank you so much, John. I'm very happy to have been invited here, and it's going to be very hard to follow Christine Jones. Thank you very much. And I'm going to share my screen now and try to get us started. Okay, is it shared? You see it?
Yes. Yep.
Okay, so what I'm going to try to convince you of is that plants are cultivating, or I use the word farming, soil microbes and endophytes in their roots and just outside their roots in the rhizosphere. So like humans in farming plants, crops, plants themselves are farming microbes. And okay, so just consider a plant like hemp or marijuana. If you take a seed of hemp, just a naturally produced seed, germinate that, and then examine the roots. What you're going to find is that if you look over here to the image to the right, you can actually see a root hair. And you can see these spherical structures inside those root hairs. So those spherical structures are actually bacterial endophytes that this seedling has taken into its roots. So every plant has these kind of endophytes. Every plant farms microbes at some part in their life cycle outside the tips of their root cells, outside the tips of their roots in the rhizosphere, and takes them into the roots. And okay, so cannabis is just one example. Here's another example. This is a big cactus called a caduceus cactus that occurs on the desert island of Bonaire in the Dutch Antilles. And you can see these cacti are huge, but they produce fruits up near the top, these orange fruits that look kind of like the opuntia, the regular flat cactus that you see a lot on the ground, very low cacti, but similar kinds of fruits. You take the seeds out of those fruits and they look like this. And if you take those seeds and germinate them, you get a seedling that looks like this. This seedling was actually stained with a stain so that we could visualize the bacteria inside it. And that's why these roots actually are orange in color, because this stain we use causes the microbes around the microbes to change color, change an orange, reddish, brown color. So this is the root hair of several root hairs here laying on this image here. And this is the caduceus cactus, and you can see inside all these little dots. All those dots are microbes or bacteria that this cactus has taken into the roots. So happens that plants will carry some of these microbes with them on the seeds and or in the seeds, and they'll be in the seedling when it germinates. But the plants will also attract microbes from the soil and then bring them into the root and process them inside the root. So, but once they're inside, we can call them endophytes. And endophyte, I should define that a little bit better. An endophyte is any microbe, bacterial or fungal, that goes inside the tissues of plants but doesn't cause disease. So they're not pathogens, but they have these other beneficial effects on plants. And these endophytes can come from the soil, they can come from the seed. In fact, they come from both. So, in terms of agriculture and growing plants without chemicals, it's necessary to have a very healthy soil with lots of microbe diversity in it, nutrients in terms of organic forms of nutrients and other forms of nutrients, in addition to seeds that contain your microbes, so seeds that you have not sterilized in any way or have not removed coatings, because many times these microbes are vectored on seed coatings. So it's important to have seeds that have the microbes on them. And I'm going to jump back again. These arrows actually indicate, this is a root hair again, these arrows indicate the bacteria in the hair. And you can see they're in pairs here in places, the arrows indicating that. That's where they've recently divided. And so these microbes are actually replicating inside the plant, inside the root cell, and in this case, inside the root hair cell. And I'll explain more of that as I describe the process. The microbial endophytes have some effects on plants. And among the effects are that they will improve, when they're in the plant, they improve the oxidative stress tolerance of the plant. That means, for example, heat tolerance, that's an oxidative stress. Heavy metal tolerance, that's an oxidative stress. Salt tolerance, for example, high salt is an oxidative stress. And so it improves, these endophytes will improve all of those stresses, biotic and abiotic, actually. They will also suppress pathogenic fungi. So a lot of these bacteria will go out into the soil from the roots, and then they will colonize potential fungal pathogens in the soil, and they'll make those pathogens avirulent, so they won't cause disease. And I'll mention that again near the end of the presentation. These endophytes also modulate or control root development. That is to say, when the endophytes are present, roots will show proper gravitropic response. That is, they will grow downward, and they will also produce root hairs. Okay, but if we remove the microbes by very rigorous surface sterilization and antibiotics in some cases, the gravitropic response is lost. Roots will grow only on the surface. In some cases, the roots will go into the air, but they won't grow down. And also, roots will not have root hairs. So those root hair formation is a function of having those microbes present inside the root cells. Okay, the microbes present will improve nutrient function. That's actually the function of this rhizophagy cycle that I'm going to describe for you, and involving these microbes, these endophytic microbes. And that is, these microbes will bring nutrients into the plant, and then the plant will extract from those microbes the nutrients so that the plant can then acquire those nutrients. And finally, I'm going to mention and explain how these endophytes, particular endophytes, alter the chemical constituents of plants and may make crop plants more healthful or less healthful, or less full of antioxidants versus containing antioxidants. So I'll explain that. The phenomenon where roots were taking in microbes was actually noticed first by some Australian investigators, in Queensland, Australia. And this is one of the investigators, Channy Pengfu, of that group, one of the young investigators. She was a young investigator there in that group. Several people there involved in that work. But what they found was that plants are taking in the yeasts and bacteria into their root cells. And then in time, those microbes are being degraded. At least some of them are being degraded. And what they did actually is they labeled the yeast and the bacteria with green fluorescent protein so they could track it, so they could see it inside the plant. Then they fed it to tomato and they fed it to Arabidopsis plants, and then they tracked it using fluorescent microscopy. And you can actually see here, this is from their paper, and you can see at D here, you can see those are actually root hairs, and you can see all these dots, fluorescing dots in there. Those are all the microbes inside those hairs or inside those root cells. And if you go to E, you can see there is actually there's a root hair there, and you can see all those fluorescing dots. Those are yeasts inside those root hairs. In time, the fluorescence dissipated, and these investigators proposed that what's going on here is that plants are actually internalizing and degrading microbes. So they are essentially eating microbes in the soil. And they published their paper in a journal called PLOS One, an important open access journal that we use frequently. And they titled it "Turning the Table: Plants Consume Microbes as a Source of Nutrients." And this was published in 2010. Later on, they named this process rhizophagy. Rhizo meaning root, phagy meaning eating. And that is to convey the idea that this is roots eating microbes. And our contribution, we were working concurrently on this, but we didn't actually realize the microbes were inside the roots at the root cells were being degraded. But we began to look inside, and in fact, we saw that those microbes are going into the roots. In fact, we saw this process as more of a cyclic process, and so we described the rhizophagy cycle, cyclic. And what's happening in the rhizophagy cycle is these microbes are going into the root cells at the root tip. So the root tip is actually secreting exudates. So it's sugars, organic acids, and some amino acids, and some other nutrients there in order to attract microbes from the soil and to cultivate those microbes right outside that root tip, right just around the meristem where the cell walls are soft. And those microbes are actually taken into the root cells at that place where the plant is cultivating them at the root tips. And I'll show you a little how that happens a little more in detail. But the microbes are taken in. We don't exactly know how. That part we don't understand, but we can see them going in, and we can see them once they're inside there. Once they're inside the root cell, they actually go into a space called the periplasmic space, which is underneath the cell wall, but not quite inside the root cell itself. So it's really just outside that root cell plasma membrane. I'll show you that. But the root will then secrete superoxide, reactive oxygen superoxide, which is very, very potent form of oxygen that will oxidize the cell walls off of the microbes. And so those microbes, those bacteria that go in, or the yeast that go in, will get stripped of their cell walls, and then they become these naked protoplasts, and then they're actually managed internally in those root cells by the root cells, moving them around. They extract nitrogen from them, or not only nitrogen, other nutrients as well, and I'll explain that. But they make the plasma membrane of the microbe leaky with that superoxide, and so nutrients go into the plant out of the microbe, and they can then the root cell absorbs it. The surviving microbes, some of those microbes are broken down completely. Other microbes survive, and the ones that survive actually are replicated inside the root. It's really fascinating. The root clones them, breaks them into many, many pieces, and then clones them. From the few that go in, many, many more come out. What happens is they will accumulate in these root hair initials and on the root epidermis. So right around where the root hairs form, the microbes are in those initials. And the microbes present there in the root hair initial causes the root hair to elongate. If we remove all the microbes, we have no root hair elongation. You have no root hairs. So the microbes are necessary to have root hair development. And as those root hairs develop, those microbes are shot out of the tips of those root hairs. They are essentially ejected from the tips of root hairs. The root hairs elongate, and they eject the microbes as they elongate. And then once they're ejected from the root hair, the bacteria will reform their cell walls. They'll reform flagella, whatever they need to swim, free living in the environment. And then they will go back out into the soil and acquire more nutrients, and then be attracted back again to the root tip to bring those nutrients back to the plant, and then go in. So the plant will actually colonize the soil with those microbes that work well in the rhizophagy cycle and that function in the rhizophagy cycle. So there's an image here to the right top, and you can see a cell there that actually is a root cell, and you can see the bacteria inside it. That's where they go in. When they go in, they'll have their cell wall still. You look at C over here, and you see a root tip, and that ejecting the microbes outside that tip, and there you can see the bacteria coming out. Okay, so this is a grass seed. Grass seeds will carry several species on the seed, and when the seed germinates, they'll come out and they'll colonize the root and they'll colonize the shoot. But the rhizophagy cycle happens only on the root. So colonization of the root is what we're going to focus on. So this is just a diagram of a root, and you can see the hairs up here, and you can see the root exudates there. That's where the microorganisms are cultivated. All roots will do this. All roots will make exudates, and they will cultivate microbes. And at some point, most plants, if they're able to, will form root hairs, most of them. This actually shows a grass root tip, and you can see the microbes outside there in this cloud of blue. The microbes are entering the root cells right there at the epidermis, and they go into the cells, and then the plant secretes, the root cell secretes superoxide onto them. And the superoxide is produced on the root cell plasma membrane. And it's produced by some cells called NOx, or some enzymes called NOx enzymes. And that takes molecular oxygen and makes superoxide. And that's how the plant will actually manage these and knock the cell walls off of those microbes. And this shows some bacteria. When it's hit with superoxide, you can see their rods here to the left. They have the walls hit with superoxide, and then they lose their walls and they form these little protoplast stages that divide very rapidly. You can see to the right that picture, protoplast stages. And that's how they look inside, as those protoplasts, spherical. This shows some. This is a Phragmites root. You can see chains of those microbes there. And some of those, the bigger ones are being broken down, the smaller ones, but you see smaller ones there. You can see the arrow there. You can see a chain of them with the smaller ones forming. This is a diagram that shows that. The microbe, the gray here is the cell wall of the root cell. The white is the cytoplasm of the root cell. The bacterium is between that cell wall and the cytoplasm, where the membrane actually is just that outer layer. The plant secretes superoxide onto the microbe in that periplasmic space. That where it's located is called the periplasmic space between the cell wall and the membrane. But the plant cells also move those microbes around in what we call cyclosis. And you can see these arrows that I have internally. Okay, so these cells' contents are in constant motion, and these bacteria outside in the periplasm are in constant motion. The plant is moving them around, keeping them off their toes, hitting them with superoxide, breaking them up, replicating them, cloning them essentially. Okay, this shows a root hair cell. This is a tall fescue, actually, root hair. And you can see it's stained NBT to visualize superoxide. You can see the purple or the blue, which all where the superoxide is, the little spherical things. Those are all the bacterial protoplasts inside that root cell. Okay, and I already mentioned, I won't go to much more but the bacteria necessary for proper development. You get no root hairs, you get no gravitropic response unless those microbes are inside those roots. This is a Bermuda grass seedling where the microbes have been removed, and you can see there's no hairs there. Okay, the same Bermuda grass the same experiment in other seedlings where we put the microbes back on. Now you see root hairs form immediately. Okay, so the root hairs are the microbes are necessary for the root hairs. This is near the meristem, where the microbes are being taken in to the root, near the root tip meristem. And you see in here, this white arrows are indicating little rods, bacterial rods, inside those root cells. And you can see the root cells are square, they're small. That's how they look after they just divided. And so they're in there. When they first go in, they have the walls, and you can see them. We can see the shapes then. But right after that, after they hit with superoxide, they lose those cell walls. This is a bit older. You can see the root, all those little brown dots. And those brown dots, you can see the hairs here. All those brown dots in there, all microbes. And so if we look close at that, this is a hair. You can see all these dots in here. All these are the microbes. These are all the protoplasm of the microbes. These hairs become engorged with microbes. They're processing microbes. They're extracting nutrients from these microbes. Some of them are being digested and degraded. They're getting nutrients out of these microbes, and they're replicating those microbes. We call the process where they will eject the microbes from the hairs. We call that cyclosis expansion wave mechanism for microbe ejection from hairs. And this is just a diagram. It shows a hair. And you can see the outer line on the outside. The image to the left shows the tip of the hair, and you can see the outer line is actually the cell wall of the hair. And the cell wall is thick lower in the hair, but it's thin right at the tip. It's elastic at that tip. And there's constant cyclosis. That is, there is movement. You can see the arrows here, movement of those microbes, moving them around, conveying them around like a conveyor belt, pushing them to the tip and occasionally catching some and moving them back and so forth, but also hitting them with superoxide to get nutrients from them. And here at the tip, hitting them with superoxide. The actual root hair itself is not all the way out to that wall. Instead, the root hair, if you see the dotted line inside, that's the root hair membrane. So it's actually a needle shape inside that root hair, inside that wall. It's a needle, the root hair. And so all of this area at the tip where those dots are, that's all microbes out there. So the whole fatness of the tips of these hairs, the whole width is actually microbes there, with just a little bit having to do with the root hair itself inside. Then what happens is that there is an expansion wave that starts at the base of the hair and it goes to the tip. And as it goes to the tip, it will push those microbes out to the sides and then move to the tip and push them out of pores that form in the tip, little holes. And then once those microbes are out, they'll reform the cell walls and they'll make flagella, and then they'll swim out and acquire more nutrients. As soon as they no longer are exposed to superoxide, they reform the cell walls and they become competent. Also competent to live in the soil. And also the other thing is the root hairs will secrete nutrients out after those microbes so that those microbes have some nutrients, some carbon, some sugars to rebuild their walls. Okay, this is one of these microbes one of the microbes, one of the plants that we studied. This is a sedge on the desert island of Bonaire, and it actually doesn't have much soil. It grows on these rocks and these little pits and rocks and also on sand. But what it does have is a lot of microbes. And this actually, the lower image shows one of the root hairs of this sedge, and it's stained for reactive oxygen. This is a stain called diaminobenzidine. It stains red and brown. I talked about that stain before. I didn't mention it, but told you we used that. And inside this area is actually where the microbes are. So the plant is secreting reactive oxygen onto the microbes, and that's why we're able to see them. We can with this stain, this diaminobenzidine, this DAB, we can see hydrogen peroxide. And so there's hydrogen peroxide around those microbes. If you look in there carefully, you can see the blue in there. That's actually the microbe cytoplasm itself. So the staining is right around. Okay, and if you look at the top, this top is a movie that we made through the reverse of a Petri dish. And you can see now those microbes are in constant motion. That's the cyclosis, the movement of those microbes around like that. That cyclosis is important because what happens is the plant is breaking down gradients that form, nutrient flow gradients that form between the microbe and the root cell itself. If this weren't moving like this, if cyclosis weren't happening, gradients would build up, and that would slow the transfer of nutrients from the microbe to the plant. And also, I should say the other direction as well. So with those gradients broken down, the nutrients flow very rapidly between the microbe and the plant. So that movement, that cyclosis is very important in this mechanism of nutrient exchange and also breaking up of the microbes into smaller units. Okay, this actually shows one of these root hairs, and you can see a lot of a cluster of microbes there, some big, some little. Those probably started from one cell or maybe a couple of cells, and now you have a bunch there. That's because the plant is replicating, cloning those microbes inside those roots. So a few go into the very tiny uh, cells at the root tip, and many more are formed inside these developing roots. This actually shows the sedge again, and you can see the hairs the microbes moving along the hair, and you can see them accumulating out the tip, right at the tip. And that accumulation at the tip is important because that's where they're going to be ejected out of, but also that's where the signal is going to come from for root hair elongation to make the root hairs elongate. And this actually shows a picture of one of those. This root hair, again, the sedge root hair. You see microbes in there, but actually you don't see a lot of microbes here. This is full. They're not all staining. The fatness of this root hair is due to how engorged this hair is with bacteria. So these hairs just get filled with bacteria. You can see they're coming out here, and as they come out, you can see some of them have rods over here where they're reforming their rods, reforming their walls and their rod shapes, and then they'll go back out into the soil. So, something like this is somewhat interesting, but this is really convincing when you do see it. And this is tomato, the young lady that did this experiment, but you see tomato root hairs, and you can see the swelling, the expansion wave beginning at the tip over here at 1, all the way to the left. You see it starting, it goes so far. And you see it 2, it's a little bit further up. You see it expanding, and you can see the microbes being squeezed out of pores at the tip. And 3, you really see a bunch of them coming out. And if you look at 3, if you look down there as it does it, you could see like 3 pores, little pores. So these microbes, protoplasts, they're malleable, and they can go right through those pores. So the pores don't have to be big, they're tiny pores. Our hypothesis for explaining how the microbes trigger root hair elongation is what we call the nitric oxide hypothesis. And that is the idea that microbial produced nitric oxide triggers root hair growth. And that is, this nitric oxide is a hormone, is a plant hormone. And it's a microbial hormone, but it's a plant hormone. And what happens when you have the microbes in the root tip all the way to the left here, you can see the little root hair tip. See all the dark area, that's the microbes. They're secreting they're secreting nitric oxide. And nitric oxide is received by the plant as a signal for elongation. And so then the root hair will elongate. So nitric oxide is a plant hormone. And what we think is that the microbes are actually secreting the nitric oxide as a self-defense mechanism, because nitric oxide acts as an antioxidant and will prevent some of the plant-produced superoxide from destroying the microbes. So the microbes are defending themselves with nitric oxide, but that's giving the signal to the plant to elongate. And I show a little chemical reaction here. Nitric oxide essentially neutralizes the superoxide. Nitric oxide plus superoxide, and you get nitrate as a product. Now, interestingly, nitrate is one of the forms of nitrogen that plants absorb and can get nitrogen from. So that's an interesting possible source of nitrogen from these microbes. So experiment done by a student of mine who tested that. This is Ivy Chang. And what she did is she removed the microbes off of plants, off of some grasses, put the microbes back on, and you can see the hairs form over here with the microbes back on. Okay, that's good. But then she stained for nitric oxide to see if those microbes are secreting nitric oxide. And this just describes that experiment. And this shows it now with the fluorescent stain for nitric oxide. And with this nitric oxide stain, you can see now the bacteria inside those hairs. And you can see they're staining for nitric oxide. So they are sources. These microbes are sources of nitric oxide. That's consistent with our hypothesis. Here's another one that shows root hair again, and you can see the microbes accumulating in the tip, and you can see they're a big source of nitric oxide. And we can also use inhibitors of nitric oxide, like methylene blue. We put it in the plants in the medium where we put the plants in, and then the hairs don't form. So we can inhibit this by inhibiting or scavenging. Methylene blue actually is not an inhibitor of nitric oxide; it's a scavenger of nitric oxide. So we scavenge it and we eliminate the growth of the root hairs. Okay, so what happens to plants without these endophytic microbes, without the rhizophagy cycle? This is rice, and to the left we have microbes in there. You can see the how big the seedlings are. Okay, so what we did is we took all the microbes off the rice and then put some back on, and you see there they become endophytic and the plants grew. Over here we removed all the microbes and left them off, and you can see those plants are actually repressed. Okay, and this is what they look like a little bit later. You can see with E plus with the microbes to the left, and then without the microbes to the right. And you can see with the microbes, look at all the soil adhering to the root. There's a whole different relationship with the soil when these microbes are present. And that's necessary. The microbes are, of course, going to the soil and bringing nutrients back from the soil. So the microbes there change that relationship to the soil. Here's without. You see there's little soil adhering. There's also no root hairs or very short root hairs in those seedlings. Plants suffer if they don't have this healthy microbial community that they can interface with. And this is all about nutrients. And very quickly, I'll just describe a quick experiment in which a visiting scientist, Shuai Xiao here, removed all the microbes off wheat, and then isolated those microbes, and then put them on to wheat one at a time, and then measured nutrients absorbed into the wheat. Okay, and this shows wheat actually grown to the left with no bacteria, so all their endophytes were removed, and then to the right with bacteria put back in, basically one at a time. And you can see to the left the plants are stunted, they're poor, didn't grow very well, they're malnourished, they're small. To the right they look much better. This is a bacillus endophyte from the naturally on the wheat, but they were put back on. And you can see big roots, same age, big shoots. Plants are much better nourished. And when we look at the nutrients themselves, if you look at nitrogen, for example, to the left, the bar to the left shows the amount of nitrogen that came in without bacteria. So some nitrogen did come in. But then you look when you put the bacteria back on, it looks like about 30% more nitrogen. This seems to be a consistent number that we see for nitrogen, about 30% more with the microbes in several experiments. Phosphorus looks like in this experiment it looks like 20% more, something like that. And there are, the three bars here are three different strains of endophytes that came off of the wheat. And to the left is with none. Here's potassium. With potassium, you see no bacteria way down here. And then with one of the bacteria, you can see way up here. It looks like 3 times the amount of potassium, huge amount more potassium. With the other bacterium, you can see it's like double at least, and the fourth one, this fourth bar, double again. So a lot of other iron is impacted, zinc is impacted, magnesium is impacted. Many nutrients are impacted by these microbes, and microbes carry different nutrients. They're not all the same. Microbes are different. So really, plants will need a whole community of microbes in order to get all the nutrients that they need. And we don't know how plants select. They may be selecting to maximize the nutrients. Okay, so strains of bacteria are unique in terms of the nutrients they provide the plant. Okay, but those bacteria also make the plant more alter the chemical constituents, like for example carotenoids. And we did an experiment there where we put 3 different bacteria from other plants onto carrots and then measured the carotenoids. And this is actually in carrot root. You see the root here, you see the little dots in there, those are the bacteria inside there. And you can see the root, you can see all these little spherical things. That's the bacteria inside the root cells. And here's for beta carotenes. And beta carotenes, the control without bacteria, you can see over here, the slow bar to the left. And then you see with one of the bacterium 1, you see it didn't have any effect. That bacterium didn't seem to help at all. But then you have bacterium 2, and look, it's at least double the amount of beta carotenes that are present. Bacterium 3 improved. So different microbes have different properties. And these microbes, for plants to do the rhizophagy cycle and have these microbes makes them healthy for different reasons. One is because they get nutrients from the soil. That's important. They get the rhizophagy cycle allows them to get those nutrients right out of the soil microbes. But also because it causes them to have increased oxidative stress tolerance. Because they're using reactive oxygen to get those nutrients out of the microbes and to control those microbes, they themselves, the plant themselves, upregulate their antioxidant genes and their oxidative stress tolerance genes and so forth. So then if another oxidative stress comes along, like, for example, salt or drought or heavy metals or whatever they are, the plant is already resistant to it. And third, because those microbes are interacting with other soil microbes, with soil pathogens, the bacteria will go out and they'll colonize soil pathogens. They reduce the virulence of those other soil pathogenic fungi, and those fungi are not pathogenic, and so the plant is not affected by disease. So really, that's the. I know I'm out of time, so that's my conclusion for this presentation. Here are some references for work that this work was published in. These are open access articles, and you can get these. One published in 2018 and another in 2019. Freely available online. And these are people, students in my lab and visiting scientists and other people who contributed to this work. It took a lot of people to do this work, and they all deserve credit for what they've done. And that is it. And do I have any time for questions, John?
Yes, yes. Well, first of all, thank you very much, James. That's very comprehensive indeed. And as I say, the perfect addendum to Christine's earlier presentation. I think the two fit together really, really well. Yes, we've quite a lot of questions in actually. So people have been paying close attention. We're going to start with Brian Murphy, and Brian says, James, is there any benefit to identifying soil microbes down to genus or species levels, or is that overkill?
Well, I should say I know Brian Murphy's work for years. It's very important work that he and his collaborators are doing. I tell you, it's a good question. We have the tendency is for investigators to try to look at the minutiae of all those microbes, and for us, we needed to simplify, and so what we have tried to do is not to dwell on all the soil microbes. Instead, what we've tried to do is look at those that are vectoring in and on the seeds. And the reason for that is that it is a hypothesis, but our idea here is that plants have selected the best microbes and from the soil, and that they have then placed those microbes in and on their seeds so that their seedlings could better survive. And all the work that we've done suggests that's correct. That's a correct assumption. But I have to tell you, we are working mostly with seedlings. And so if you're growing a crop in biodynamic conditions, you're getting a lot more benefit from those microbes than just what's seed vectored. You're getting all of those microbes out in your soil that you're cultivating, and some of those microbes are being actually taken into the plant, and then they will end up on the seeds of those plants again. But that's too complicated for us to dissect. I should also add that many microbes have been lost from seeds through our cultivation process, and so the efforts by Brian Murphy and other people to find microbes that they can put back with plants that would then enable those plants to grow better is actually, in my opinion, in my studies, is compensating for where we've lost microbes. So it's reacquiring microbes that have been lost.
That's interesting. So you can actually replenish the soil by reintroducing microbes and improving that relationship between plants and microbes, even where they've been lost. Yeah.
You can. In fact, there's certain biostimulant microbes that you could get, even commercially, and take those and put them on, and they will help. Now, not all biostimulant microbes are the same. They're not all equal. Some are much better, and I can tell you that you could select, if you're selecting for seed vectored microbes, you may end up with better microbes than if you just go out for soil microbes.
Okay. I'll move on, James, if I may. We have a question from David Bird. And David asks, how damaging are seed handling systems like augers, conveyors, and so on to the microbes on seeds? And he also wants to ask whether seed treatments to the seed crop affect the natural biology on the seed that is produced.
I mean, that's really a good question, David. Yes, I'll try to answer the second part first, and that is seed treatment. I mean, the worst treatment is, for example, how we treat cotton. You take cotton seeds, we drip it in sulfuric acid or some acid to remove all the lint, all the little fibers off of it, and we actually we eat the fibers, but we also remove all the microbes and destroy those microbes. And then those cotton seeds are then, and we do that to go through the planter. Those cotton seeds are then have no microbes there. They are sitting ducks. So they go into the soil, anything attacks them and kills them. They don't have their defensive microbes. They don't have their microbes to function in the rhizophagy cycle. They're in bad shape, though. So we put chemicals all over them to grow cotton. That's the only thing we can do. Take, for example, I'll give you another example. Just the cultivation, the act of cultivation will remove some microbes. There was a study a number of years ago with the wild tobacco, an annual tobacco, in which some investigators in Utah took this wild tobacco. They put it in cultivation every year. They would take it, bring it inside, and store the seed, and then the next spring plant it again, and take those seeds and bring it inside to a dry place. That's how we cultivate, right? So 7 years they did that, and then they got this wilt disease that started wiping out their populations. They went back to the wild populations. They got their microbes off of that, and they put them back on their seeds, and voila, the end of the epidemic. American Indians did something interesting. American Indians would growing corn. They cultivated corn for a long time, but they would take their corn, and before they planted it, they would use what they called the corn medicine. And this corn medicine, what they would do is they would take wild roots of grasses like Hystrix, Patula, Phragmites, the native Phragmites that we have here and less here. Take the roots, put it in water, maybe heat the water a little bit, get the microbes out, and then they would germinate their corn seeds in that water. Once they partially germinated, then they take the corn and plant it. Okay, so they were taking these wild microbes, these wild plants, getting the microbes off the roots. They were putting it on the corn again and planting the corn and essentially replacing those microbes that had been lost in a crop that had been cultivated a long, long time. Okay, now the planters, people will sometimes remove husks, for example, in certain grasses, that the microbes are carried on the husk. They'll coat seeds with clay sometimes. We don't know if that's a negative effect or not. I think if it's not hurting the microbial community, then it should be okay. If you put fungicides there, we're only putting fungicides there because we've damaged the microbes, for one, and they can't then protect from diseases. I do think the answer to both of those parts is yes. We're affected by planters, how we treat seeds for planters, and other seed treatments. Yeah,
it's very interesting, James, what you say about the Native Americans figuring out, if you like, through practice and custom, what science has since actually given us a methodology to understand why that was beneficial. So that's a really, really interesting point. I'll move on to Jack Healy, who asked the question, do you have any thoughts about cheap forms of myco-remediation for riparian zones, example, to improve water quality?
Well, I do know that there that there is a lot of work. I am a mycologist. I actually have got 2 mycology courses going on this semester, so I've got students during this pandemic running around identifying mushrooms, the woods, scavenging the woods of New Jersey, identifying mushrooms and studying other fungi. So, yeah. So,
can I ask, by the way, at this stage, do people think you're a fun guy? Sorry, you've never heard that one before, but do go ahead.
Yeah, right. I've never heard that before. I've never heard that, John. You threw me off with that one.
Okay, so right would you like the question again?
No, I know it. You don't have to. I do it. I just as soon as I saw Jack Healy's name again, I remembered the question. Okay, so there are people who are exploring this water remediation, cleaning water, and also soil remediation using fungi. And I think you'll find online reports of how people are doing that. We, in my lab, we haven't done that directly, but I have students on a regular basis giving presentations about other groups that have done that, how they use. In fact, last night we just had a student talking about it. So I don't have any great answers, except that Paul Stamets, I think, has been involved with some of that, and you might be able to go on his website. And I also had a student, one of the graduate students from here, that did a program, had a grant, I think, to look at that at one of the Midwestern colleges. But I can't give you too many details now.
Yeah,
I appreciate the question.
Sure. I'm going to hand you over to Sean. I think you've got a question for James.
Yeah, James, we've actually loads of questions, but we're running late, so we're not going to get through them all. But there was one interesting one here from Stephen. Can GMO crops exchange microbial endophytes with weeds and thus transfer engineered tolerance traits to those weeds?
So yeah, that's interesting. The GMO crops, their own genomes have been modified, and can microbes then move into the GMO plants and then move over to the weeds, or the other way around? The reason I say is because many of these GMO crops have gone through a tissue culture phase, and that really cleans off the seed vectored microbes, at least. So whatever they get new, and they do get stuff new, it's from the soils. And if they have good soils, if it's a biodynamic soil, they could be recovered and get good microbes. If it's a depauperate soil, they could get only a few microbes, and they can get pathogens. They're more susceptible to pathogens. Will those microbes vector the genes from the GMOs to the weeds? I don't think so, but there always is. I never say never in biology. Never say never, because it is certainly possible. We do know that lateral gene transfer has occurred between organisms many, many times. Okay. So it is possible.
Right.
One last question.
Yeah, okay. I'll leave the last question then to Glenn Boyd. And again, Christine referred to glyphosate earlier, so this is specifically on that. And Glenn asks, does glyphosate affect the root hair's ability in absorbing nutrients or micronutrients from the microbes?
So it should affect it, particularly because glyphosate is also antibacterial, and it has been shown to inhibit some bacteria. Other bacteria can break down glyphosate. We really don't know all the effects of glyphosate. I mean, this is something people have been asking us for a couple of years now, so it's a really good question. We have not been able to answer it just because we're trying to answer some other questions. And, the pandemic also slows us down a little bit. So we haven't, but the expectation is, though, yes, it will impact the ability of plants to acquire nutrients from microbes. So yes, it's affirmative, that answer to your question.
Okay. As a final thought, when you're looking down the microscope, when you're looking at healthy soil, when you're looking for healthy soil, what do you expect to see down the microscope? What tells you this is a healthy soil sample I'm looking at?
I would look at the seedling themselves. You know, what microbes are the seedlings taking from the soil? So I would filter the question. You know, what would tell me if I looked at the soil? I would look for high diversity of microbes. I would look for organic material where these microbes could feed, what they're feeding on, breaking down. Humic substances are important because that reflects the breakdown of organic material by the microbes. And then, you know, are the plants able to acquire microbes from that soil, a good diversity of microbes in the soil? So that's what I would look at.
Great. That sounds like a good place to leave it, James. And thank you again from coming to us from New Jersey. And I think that concludes this session. Thank you again, James.
It's my pleasure. Thank you so much. I really enjoyed it.
Thanks, James.
Delighted to have you.
And we're back.